The Constitution of India, Preamble. We, the people of India, having solemnly resolved to constitute India into a sovereign, socialist, secular, democratic republic and to secure to all its citizens justice, social, economic and political, liberty of thought, expression, belief, faith and worship, equality of status and of opportunity, and to promote among them all fraternity, assuring the dignity of the individual and the unity and integrity of the nation. In our Constituent Assembly, this 26th day of November 1949, do hereby adopt, enact and give to ourselves this Constitution.
Hi students, welcome back to Zoology class. Hope all are fine and safe at home. Today I would like to start the class with the words of the famous scientist Albert Einstein. If you want to live a happy life, tie your life to a goal, not to a person or a thing. I'll repeat once more. If you want to live a happy life, tie your life to a goal, not to a person or a thing. Success is the way to happiness. If you don't have any aim or goal in your life, there won't be any success. That means there won't be any happiness. So everyone should have an aim or goal in their life. It will make their life more meaningful. Okay. Now let's move to the topic. In the last chapter, we have learned about the identifying features of a living organism and what is the importance of classification, how can we classify organism, which are the different levels of classification, taxonomic hierarchy, and different branches of science which involve in the process of classification, isn't it? So now we are going to study about the taxonomical aid that can be used in the identification and classification of organism. Okay, what is meant by taxonomical aids? Taxonomical aids are the procedures and the techniques followed by the biologists to identify and classify an organism into different taxonomical level. Can you say the importance of this taxonomical aid? Yes, taxonomical aids are very essential in taxonomical studies. Taxonomical studies of various species are essential in different fields like agriculture, forestry and industry, etc. And these taxonomical studies require the correct classification and identification. This classification and identification is possible only if there is an enough availability of the specimens. So there is a need of different techniques and procedures to store and preserve the specimens for future use. Isn't it? Taxonomical aids plays an important role in the storing and preservation of different plant and animal species. You might have understood the importance of taxonomical aid, isn't it? Without taxonomical aid, it is not possible to classify any organism under suitable category. That is the relevance of taxonomical aids. There are different kinds of taxonomical aids. For example, herbarium, botanical gardens, zoos, museums, keys, flora, manuals. Like this, many taxonomical aids are available. Among these taxonomical aids, we are going to study about herbarium first. Okay, first we are going to study about herbarium. As I told you earlier, herbarium has an important role in the identification and classification of different plant species. Okay, actually this herbarium is the storehouse of collected plant specimens. Here, different plants are collected, dried and pressed in herbarium sheets. Okay, and these sheets are arranged on the basis of a universally accepted system of classification. Actually this herbarium is a storehouse where we can see different kinds of plant specimen in herbarium sheets, and this herbarium sheet can be kept for a long time and can be used for a future purpose. Okay, and here in the case of a typical herbarium sheet, a single herbarium sheet may contain the plant specimen in its pressed form. Along with it, it contains a label that carries necessary information about the plant specimen. For example, label may contain the place of collection and the date of collection of that plant, and collector's name, botanical name and local name of that plant. അതായത് collector's name and from where he got that specimen, in which date he got that specimen, what is the local name of that plant, what is the botanical name of the plant, which is the family of that plant. Such kind of basic information will be there in that label, which can be found along with the herbarium sheet. You will study the preparation of herbarium sheets in botany in higher classes. So at that time you will become more familiar with this. In short, herbarium sheet will act as a reference material to identify and classify organism under suitable category. Similarly, botanical garden is another important taxonomical aid which can be used to identify plant of various species. Okay, here botanical garden contain a large number of living plants in it. അതായത് ഇന്ന് വംശനാശ ഭീഷണി നേരിടുന്ന പല പ്ലാന്റ് വെറൈറ്റീസും നമുക്കവിടെ പ്രൊട്ടക്റ്റഡ് എൻവയോൺമെൻറ്റിൽ കാണാനായിട്ട് പറ്റും. The plants which are there in the botanical gardens are labeled with their botanical name as well as local name. So it will be easy for the people to identify the plant specimen very easily. There are many botanical garden in India. Among that, Indian Botanical Garden located at Howrah as well as National Botanical Research Institute located in the Lucknow are the two important famous botanical gardens of India. The world's famous botanical gardens are located at Kew in England. So museum is another important taxonomical aid that can be used to identify plant as well as animal specimen. You can say that museums are the storehouse of plant as well as animal specimens. These kind of museums can be found associated with the schools and colleges, and the mode of preservation which we are adopting in the museum will be different from that of the other taxonomical aids. അതായത് നമ്മൾ ബാക്കിയുള്ള ടാക്സോണമിക്കൽ എയ്ഡ്സിൽ ഉപയോഗിക്കുന്ന മോഡ് ഓഫ് പ്രിസർവേഷൻ അല്ല ഇവിടെ എന്ന് പറയുമ്പോൾ, ഇവിടെ മ്യൂസിയത്തിൽ എപ്പോഴും ഡെഡ് ആയിട്ടുള്ള ആനിമൽസും പ്ലാന്റ്സും ആയിരിക്കും അവൈലബിൾ ആയിട്ടുള്ളത്. സോ ദ വേ വിച്ച് വി ആർ അഡോപ്റ്റിംഗ് ടു പ്രിസർവ് ദ സ്പെസിമെൻസ് വിൽ ബി ഡിഫറെൻറ്റ്. Animal specimens are preserved on the basis of its size. For example, if it is a small organism like fish, it is preserved inside the preservative solutions like formaldehyde. If it is large animals, it can be stuffed, okay, and it can be kept inside the containers of the museum. Okay, it can be kept for a long time within the containers. Small glass containers will be there, and inside that containers this can be preserved. And in the case of insects, their mode of preservation is different from that of the other organism, and these kind of insects are preserved inside the insect boxes. That means these are the boxes made from cardboard. Actually here the insects are collected and pinned within the boxes called the insect boxes. Means here in museum the insects are preserved inside the insect boxes. These are the specialized boxes which can be made from the cardboard. Plant specimens can be preserved for a long time in a dry manner. Okay, that means dry specimens can be preserved inside the museum for a long period. And this museum have the collection of skeletons of animals too. You would have gone to museum many times, isn't it? Where you can see different kinds of plants as well as animal specimens. The mode of preservation will be different. That means the mode of preservation is depending on the nature of that specimen. If it is a large sized mammal, we can be stuffed and preserved after removing its internal organs, and in the case of smaller organism, we can keep it inside the preservative solutions. Like organs, if it is an organ of any organism, it can also be preserved inside the preservative solutions. Preservative solutions ആയിട്ട് നമ്മൾ സാധാരണ ഉപയോഗിക്കുന്നത് എന്താണ്? നമ്മുടെ ഫോർമാൽഡിഹൈഡ് സൊല്യൂഷനാണ്. Now let's move on to the next taxonomical aid, zoological park. Here zoological park are the places where wild animals are kept in protected environment under human care. Zoological park will enable us to learn to the food habits as well as the behavioral pattern of these kind of wild organisms. Okay, and it will be helpful in identifying the wild organism which cannot be seen in our normal forest. Okay, nowadays you can see the foreign species also. Nowadays we can see the foreign animal variety in our local zoos also. Okay. Keys are one of the important taxonomical aid that can be used in the identification of different organisms. A taxonomic key can be made by considering the contrasting features of each organism. Okay, that means on the basis of the contrasting features, we can reach a conclusion about that particular organism. As I told you earlier, here in the case of key, we are making use of the contrasting features of a particular organism to identify that organism. Here, the contrasting features are used in two alternative forms. That means it can be used as a pair. Actually, these keys are made by the taxonomist. Okay, and when he make a key, at that time he will consider the contrast identifying features of a particular species or a phylum. On the base of the features, he will make a key. Okay, and here there is a typical example of a key. And here I made a key to identify the phylum Chordata, and here I used three contrasting features. That means here I used the presence of notochord as one contrasting feature, and the presence of nerve cord as another contrasting feature, and the presence of gill slit as another one. Here, based on these three basic features, we are going to find out whether this organism is chordata or non-chordata. If the first character is yes, then the second alternative will be always no. കാരണം നമ്മൾ കോൺട്രാസ്റ്റിംഗ് ഫീച്ചേഴ്സ് ആയിരിക്കും എപ്പോഴും ഒരു പേരിലെടുക്കും. ഇവിടെ യെസ് ആണ് നമുക്ക് ആൻസർ കിട്ടുന്നതെങ്കിൽ നമ്മൾ ബാക്കിയുള്ള ഫീച്ചേഴ്സും കൂടി നോക്കും ആ ഫീച്ചേഴ്സ് ഉണ്ടോ എന്നുള്ളത്. ഇനി നോ ആണ് നമുക്ക് ആൻസർ കിട്ടുന്നതെങ്കിലോ വി വിൽ ഫൈൻഡ് ഔട്ട് അനദർ കീ, അല്ലെ വി വിൽ മൂവ് ഓൺ ടു അനദർ കീ ടു ഫൈൻഡ് ഔട്ട് ദാറ്റ് ഓർഗാനിസം. Again, presence of notochord എനിക്ക് യെസ് എന്ന് കിട്ടി. അപ്പോൾ ഞാൻ അടുത്ത് എന്താ ചെയ്യുക? I have to consider the second feature, that is presence of dorsal nerve cord, whether it is present or not എന്ന് നോക്കും. If it is present, we have to mention like yes, it is present. പ്രസൻ്റ് ആണ്. ഓക്കെ, അപ്പോൾ ഇവിടെ യെസ് ആണ് ഓൾറെഡി നമുക്ക് കിട്ടിയിട്ടുള്ള ആൻസർ എങ്കിൽ ഓട്ടോമാറ്റിക്കലി അതിൻ്റെ ഓപ്പോസിറ്റ് ഫീച്ചർ ആയിരിക്കും അടുത്ത ചോയ്സ് എന്ന് പറയുമ്പോൾ. അല്ലെങ്കിൽ ഡോർസൽ ഹോളോ നെർവ് കോഡ് യെസ് എന്ന് കിട്ടിയെങ്കിൽ അടുത്ത ചോയ്സ് എപ്പോഴും നോ എന്ന് തന്നെയായിരിക്കും വരിക. ഓക്കെ, അതായത് അടുത്ത ഫീച്ചർ അതിൻ്റെ തൊട്ട് താഴെ ഓൾട്ടർനേറ്റീവ് കൊടുക്കുമ്പോൾ അവിടെ നമ്മൾ നോ എന്ന് തന്നെ മെൻഷൻ ചെയ്യണം. ഓക്കെ, എഗൈൻ പ്രസൻസ് ഓഫ് ഗിൽ സ്ലിറ്റ്സ് നോക്കുകയാണ്. ഇറ്റ് ഈസ് ഓൾസോ ദെയർ, അതായത് ഗിൽ സ്ലിറ്റ്സ് ഓൾസോ ദെയർ എന്ന് വിചാരിക്കുക. അവിടെയും ഞാൻ യെസ് എന്ന് മെൻഷൻ ചെയ്തു, അപ്പോൾ ഗിൽ സ്ലിറ്റ്സ് ഉണ്ട്. സോ ഹിയർ ഐ ഗോട്ട് 3 yes, ദാറ്റ് മീൻസ് പ്രസൻസ് ഓഫ് നോട്ടോകോഡ്, presence of dorsal tubular nerve cord, presence of gill slits. അപ്പോൾ ഫൈനലി നമുക്കിവിടെ എഴുതാൻ പറ്റും ഡ്യൂ ടു ദ പ്രസൻസ് ഓഫ് നോട്ടോകോഡ്, dorsal tubular nerve cord and gill slits, the given specimen is a chordata. ഈ കീ അവസാനിക്കുമ്പോൾ നമുക്കതിൻ്റെ മെയിൻ ഫീച്ചേഴ്സ് എല്ലാം മനസ്സിലാവും. ഈ ഫീച്ചേഴ്സ് ഉള്ളത് കൊണ്ട് the given organism will come under a chordate എന്ന് നമുക്ക് പറയാം. ഇത് ഞാൻ നിങ്ങളെ ജസ്റ്റ് ഒന്ന് മനസ്സിലാക്കി തരാൻ വേണ്ടിയിട്ട് പ്രിപ്പയർ ചെയ്തൊരു കീ ആണ്. ഇറ്റ് ഈസ് നോട്ട് ദ ഒറിജിനൽ വൺ, ഇതല്ല ഒറിജിനൽ വൺ. ലൈക്ക് വേസ് മെനി ഓൾട്ടർനേറ്റീവ്സ് വിൽ ബി ദെയർ ഇൻ ദ കീ. ഞാനിവിടെ എടുത്ത 3 കോൺട്രാസ്റ്റിംഗ് ഫീച്ചേഴ്സ് ആണെങ്കിൽ ഒരു കീയിൽ n നമ്പർ ഓഫ് കോൺട്രാസ്റ്റിംഗ് ഫീച്ചേഴ്സ് കാണും. ഈ n നമ്പറും സാറ്റിസ്ഫൈ ചെയ്താൽ മാത്രമേ നമ്മൾ ഒരു ഓർഗാനിസിനെ ഒരു പെർട്ടിക്കുലർ കാറ്റഗറിയിലേക്ക് ക്ലാസിഫൈ ചെയ്യാനായിട്ട് പറ്റുള്ളൂ. Understood? Hope you have understood what I intended. Here I have to say one more thing. When we നമ്മളൊരു കീ എടുക്കുമ്പോൾ when we consider a key, at that time I told you each feature will be there and alternatives will be there in each feature. അതായത് ഓരോ ഫീച്ചറും ഓൾട്ടർനേറ്റീവായിട്ടാണ് കൊടുത്തിട്ടുള്ളത്. അതായത് when we make a key, at that time we will make use of n number of contrasting features. Among these n number of contrasting features, a single pair of contrasting features is known as a couplet. Okay, we can call it as a couplet. Here I mentioned it. Okay, couplet. Couplet means ഇവിടെ നമ്മൾ ഒരു പെയറിനെ വിളിക്കുന്ന പേരാണ് couplet എന്ന് പറയാം. Okay, and each sentence in a key is known as a lead. ഒരു കീയിലെ ഓരോ സെൻറ്റൻസിനെയും നമ്മൾ പറയുന്ന പേരാണ് a lead. Okay, lead. Here I mentioned. Okay, a lead is a single sentence in a key and a couplet is a pair of contrasting feature that can be found in a key. ഒരു കീയിൽ കാണുന്ന ഒരു പെയർ കോൺട്രാസ്റ്റിംഗ് ഫീച്ചറിനെയാണ് നമ്മൾ എന്താ പറയുക കപ്ലെറ്റ്. അതായത് ദാറ്റ് മീൻസ് മെനി കപ്ലെറ്റ് വിൽ ബി ദെയർ ഇൻ എ കീ. There will be different keys for identifying different taxonomic levels. That means ഓരോ ടാക്സോണിനും ഓരോ കീ ആണ് ഉള്ളത്. ഓക്കെ, അതിൽ ഓരോന്നിനും സ്പെസിഫിക് കീ ഉണ്ട്. ആ കീ എടുത്ത് നമ്മൾ നോക്കിയിട്ട് വേണം കൺക്ലൂഷനിലെത്താനായിട്ട്. I'll tell you about key once more. Key is a taxonomic aid which can be used to identify organism on the basis of its contrasting features. The contrasting features can be written in an alternative manner. By comparing the contrasting features of that organism, we can reach to a conclusion about the category in which the organism belongs to. A pair of contrasting characters which can be seen in a key is known as a couplet. Each statement in key is known as lead. Okay. In addition to key, there are some other taxonomical aids. They are flora, manuals, monographs and catalogues. I'll tell you about each one within a short description. Okay. More flora will give a complete account of the habitat as well as distribution of various plant species located in an area. Okay, I'll tell you once more. Flora will give you a complete account on the habitat as well as the distribution of plant species which is located in area. Okay. Manuals will provide complete information necessary for the identification of the names of different species. Monograph will provide complete information about any one taxon. Okay, it may be a family or it may be a genus or anything like that. For example, it may provide information about a particular family only, or it may provide a complete information of a species. Okay, anyway, monograph will provide a complete information of a particular taxon. Next one is catalogs. Catalogs contain the list of publications which will be useful for taxonomists to identify different species and to describe about it. That means some journals will be there, isn't it? You might have heard about journals, and new findings related to the science will be published in these kind of journals, and the publication list of publications will be provided in the catalogs. I'll tell you once more. Catalog contain the list of publications which will be useful in the identification process. That means it will be useful for the identification for a taxonomist. ഓക്കെ, അതായത് നമ്മുടെ ടാക്സോണമിയുമായി ബന്ധപ്പെട്ട് വരുന്ന പുതിയ കണ്ടെത്തലുകൾ ഉൾപ്പെടുത്തിയിട്ടുള്ള ജേണലുകളോ അല്ലെങ്കിൽ പബ്ലിക്കേഷൻസോ ഉണ്ടെങ്കിൽ ആ പബ്ലിക്കേഷൻസിൻ്റെയും ജേണലുകളുടെ എല്ലാം വിവരങ്ങൾ അടങ്ങുന്ന കാറ്റലോഗ് എന്ന് പറയുക. ഓക്കെ, we have completed our first chapter. If you have any doubt, please contact me. Till then, bye, stay safe.